Past C chloroplast was discussed.
Okay. Mute koro. সেই প্লাস্টিন কি নেলবি shape of chloroplast in green algae. First one, cup set already discussed. Ribbon shaped already discussed. Girdle shaped discussed. Then reticulate found in Oidogonium. And it appears like this. You will get the chloroplast like this. This is the reticulate fashion. This is the reticulate in Oidogonium. You can call it reticulate chloroplast in Oidogonium. Then comes to the stellate found in Zygnema. It appears like this. Inside which is the pyrenoids. So this is the star-shaped chloroplast found in Zygnema. Next one, discoid. Found in Vaucaria. Discoid found in Vaucaria. So what is here? It appears disc-shaped. So this is the chloroplast. You can call it discoid chloroplast. Then comes to the plate leg. Mogusia. It appears like this, showing you, I am showing you like this. So this is the chloroplast. Now, in case of what is I said about the plate-like chloroplast,
Sir,
yes,
sir, is that protein in that stellate chloroplast? The
basically
red dot.
inside this is the pyroinate. Pyroinate is made of protein and starch both. Clear?
Yes, sir.
Now here you will get some plate-like chloroplast. So these are the found in Mogusia. So this is what about the different types of chloroplast. Same thing happens. And you know the photosynthetic pigments present in chlorophyll is green algae. Chlorophyll B, chlorophyll A, chlorophyll B, carotene, and xanthophyll. Chlorophyll A, chlorophyll B, carotene, and xanthophyll are the pigments present. And you know this is universal photosynthetic pigment. Chlorophyll A is the universal photosynthetic pigment. This is what about the pigments found in green algae. Chlorophyll A, chlorophyll B, carotene, and xanthophyll. After that, reproduction. Asexual. Or the asexual forms, one is zoospores, bi-zoospores. Somebody say Chlamydomonas. During favorable condition. During favorable condition, it withdraws its flagella and protoplasm gets shrinked and become round in shape. Then it undergoes the mitotic division. to produce a number of spores like this. After that, they become flagellated and they are called zoospores. When the wall is broken down, zoospores are liberated and give rise to new Chlamydomonas. As these zoospores are produced by means of mitosis, they are again called mitozoospores. They are called mitozoospores. So by zoospores, what happens here? During favorable condition, when everything is available, then shrinkage of protoplasm occurs, so that the protoplasm becomes round in shape. It starts dividing mitotically to produce a number of segments. Each one would become flagellated to form zoospores. So zoospores are motile spores. And these spores are produced during favorable condition. In favorable condition,
Sir,
this green algae, yes, bolo.
motile spore.
Motile means who
Motile
can
spore
move.
means
Motile means the spores which can move, called motile. As they have flagella, they can move, they can swim freely. That's why it is called motile spores.
okay, sir.
Two things you need to remember here regarding zoospores. One, they are produced only during favorable conditions. And secondly, they are motile spores. They can move because they are flagellated. So next one,
Sir,
yes, yes, tell me.
sir, does this happen only in the case of cup-shaped chloroplast?
Huh? No,
Sir,
I'm not getting you.
Sir, does this mitosis only occur in cup-shaped chloroplast?
No, not like that mitosis. One thing I'm telling you. Regarding zoospore formation, is the haploid plant body, in algae plant body is haploid.
Yes,
Again,
sir.
same name came. Haploid. So what happens here? It undergoes mitotic division to increase its number. And then these segments or these pieces will become flagellated to form zoospores. As these zoospores are produced by means of mitosis, they are called mitozoospores. Another type of zoospore may be found in Chlamydomonas when the zygote divides meiotically to produce spores. As such spores are called meiospores. Getting me now?
No,
Sir,
sir.
can you please repeat?
Sir,
Sir,
please
I was
repeat this part.
Sir, I was asking that is it true for all kinds of chloroplasts, or does it occur only in cup-shaped chloroplasts?
Not in chloroplast, not in an organism. Zoospores are found in many other green algae. Zoospores are found in many other green, not only in Chlamydomonas.
Sir,
Are
can
you
you
getting
repeat
me?
this part?
Zoospores are produced in many other green algae, and these spores are only produced during favorable condition. They are called mitozoospores here because these zoospores are produced by means of mitotic division of the protoplast.
Okay,
Is
sir.
it
So,
clear to you?
the shape of the chloroplast doesn't matter in the
No,
reproduction
no, no,
of the.
no. It is not related to it. Shape of the chloroplast doesn't matter here. Okay.
Yes, sir.
Okay. So these are the zoospores one form, and second one I am telling you now. Look here. Aplanospore. What are aplanospores? Produced in the beginning of unfavorable condition. When harvest started, then what happens? Protoplasm becomes round and gradually it takes the shape of a spore. You can call it a planospore. Or sometimes, maybe before the formation of juice, maybe before become flagellated, if unfavorable condition starts, then they would become aplanospores. Aplanospores are thin-walled, non-motile spore. That means they cannot move.
Sir, it will be non-motile. You have written non-motile.
Oh. Non-motile spore. So when favorable condition comes, under favorable condition, they become juice spores. To give rise to new organism. So this is what about the aplanospore. This is what about the aplanospore. Aplanospores are produced in the beginning of unfavorable condition so that they become thin-walled and they are non-motile. When favorable condition comes, they will be converted to juice spores, flagella developed and giving rise to new organism. This is what happens in case of aplanospores. Clear? So what is the difference between juice spore and aplanospores? Juice spores are produced during favorable condition.
Sir?
Aplanospores are produced in the
Sir?
beginning of unfavorable condition.
Sir,
Yes.
please repeat about the planospore.
Okay. When unfavorable condition begins, just start, then their protoplasm, it is become rounded up, become thin-walled to take the shape of a spore, and such spore is called aplanospores. Sometimes even for the formation of juice spores, some segments are developed due to mitosis, but unfavorable condition starts, then also they would not become juice spore, but will be behaving like aplanospores. Are you getting me up to this?
Yes, sir.
Is it clear? Huh?
Yes, sir.
Then what happens? So they have to overcome this period. If favorable condition comes, then these aplanospores become flagellated to form juice spores. Remember this thing, this will happen when favorable condition comes. Then this juice spore will give rise to new organism.
Sir,
Okay? Now tell me.
Sir, in this process, only one new organism is formed.
Basically, one thing, it may happen. Suppose you see, in most cases, it happens up to this. Then unfavorable condition comes and then or they can divide mitotically to produce a number of segments also. Sometimes it may be single. It depends on the condition, right?
Okay, sir.
So this is our approach. Now, if unfavorable condition continues, then what happens? You see, which one is the pronospore? It becomes thick walled to protect the inner protoplasm. So such thick walls non-motile spores are called hypnospore. They are thick wall non-motile spore. This thick wall non-motile spores is called hypnospore. Again, when favorable condition comes, under favorable condition, the thick wall structures get dissolved and these spore will form the zoospore and give rise to new organism. It is not a single like this. A lot of spores, some of these, they are all aplonospores. So they can become hypnospore if unfavorable condition continues. Because they are thin-walled, the aplonospores. They cannot resist if the favorable condition becomes unfavorable continuously. So once unfavorable condition continues, these thin-walled aplonospores are required to be converted to thick-walled hypnospore. So what is the difference between aplonospore and hypnospore? Both are non-motile. Both are produced under unfavorable condition. There are similarities, but difference is that aplonospore is thin-walled, but hypnospore is thick-walled. So these are the three types of spores I discussed here. One is aplonospore, one is zoospore, other is hypnospore. Clear up to this?
Yes, sir.
Yes, sir.
Okay. In some Chlamydomonas, what happens you see, what happens in Chlamydomonas, non-motile spores of different generations. Look, and they are found to be protected by gelatinous mass or mucilage sheath. The whole cell also protected by this mucilage sheath. You can call it mucilage sheath. Then this is called Pamela stage. Then this is called Pamela stage. What happens in the Pamela stage? The spores produced in different generations are found to be protected by the mother wall, and again they is protected by their own wall, mucilage wall, and then this is called Pamela stage. And as it is factually, then what will happen when favorable condition comes? Mucilage sheath get dissolved and all these spores are coming out. They become flagellated to form the zoospores and give rise to new organism. This is how Pamela stage is developed in Chlamydomonas, and what the consequence of these spores present inside this and when. Pamela stage is again developed during unfavorable condition, and here not only the one group of cells, a lot of group of cells are present together, protected by the mother wall, both mother wall and the cells of different generations wall found to be protected by mucilage sheath. In this way, they have to overcome the unfavorable condition. When favorable condition comes, all these mucilage sheaths get dissolved. These segments have come out. They become three types of sexual reproduction may be found here: isogamy, anisogamy, and oogamy. In isogamy, two gametes of similar size and shape takes part in the reproduction. In anisogamy, two gametes are dissimilar. In oogamy, one is very little. One gamete is very small, other is quite large. And it is sessile. Giving rise to zygote. So in isogamy, two gametes are similar in size and shape. In anisogamy, two gametes are dissimilar in size and shape. In oogamy, one gamete is very small and other gamete, which is quite large, is found to be sessile. So in this way, we are getting the zygote. Now, whatever the type, in isogamy, male and female gametes cannot be differentiated. Remember this thing. In case of isogamy, male and female gametes cannot be differentiated. In anisogamy and oogamy, male and female gamete can be differentiated. Now everywhere I am writing the term zygote. But if conjugation occurs, it seems that fertilization takes place here. What happens here? This is fertilization. So that every time we are getting the zygote. But in Spirogyra, conjugation occurs, not fertilization. I think you know what is conjugation. Conjugation refers to the union of two gametes. Conjugation refers to the whenever for the union of two gametes, gametangial union is required, then it is called conjugation. What I said, to facilitate gametic union, when for gametic union, gametangial union is required. Who is creating sound? Then it is called, when for gametic union, gametangial union is required, then it is called conjugation. So, the diploid cell conjugation is called zygospore. The diploid cell produced in conjugation is called zygospore. So this is what about the zygospore, and the product of conjugation is zygospore and product of fertilization is zygote. In conjugation, a conjugating tube is required to be developed to join two gametangia. What are gametangia? Gamete producing organs are called gametangia. Gamete producing organs are called gametangia. So this is what about the sexual reproduction in brief. Yes?
So please explain that isogamy and anisogamy part. Isogamy and anisogamy part.
Isogamy, in isogamy, two similar sized and shaped gametes take part in the reproduction. Here fusion of two similar gametes occurs. Both are which are similar in size and shape both. So you cannot differentiate which one is male gamete or which one is female gamete. Then such type is called isogamy. Is it clear now? So this is what isogamy. In an isogamy, two gametes are found to be dissimilar. As they are dissimilar, so one can be, smaller one is considered as the male gamete, a larger one is considered as the female gamete. Obviously, amount of force stored is more in the female gamete than that of the male gamete. Then this is an isogamy. In an isogamy, both the gametes may be motile, both the gametes may be non-motile, but if it is ogamy, the smaller gamete, what is called the one gamete is quite small, very, very small. So here the difference in size is much more in ogamy. So that then this is called, actually ogamy is called whenever male gamete is quite tiny and female is quite larger, then you can use the term ogamy. And ogamy is the most advanced type of reproduction. In some algae we get ogamy, in algae ogamy, but from bryophores onwards, From bryophyte, there is in bryophyte, pteridophyte, gymnosperms, angiosperms. In all these organisms, only ogamy is found. Only ogamy is found. Clear? Clear? Clear, Ajay?
Sir,
Ah,
so in ogamy, which one is motile?
male, smaller one.
Okay, sir.
I think everyone is clear now what is gametangia. So this is what in short the sexual reproduction in algae. Now I am showing you some reproductive features found in in case of conjugation. I am slightly showing you what is the conjugation, that will be easier for you. So there are two spirogyra filament. They are coming close to each other. Outgrowths are developed. In the meantime, gametes are formed. Here are gametes. Next, after that, correct conjugating tube is developed. This is called conjugating tube. Okay. Then interesting, the gametes are isogametes here. The gametes are isogametes here. So that, but one is found motile, other is non-motile. Although gametes are similar in size and shape, but in one filament, gametes are found to become motile. In other filament, they are found non-motile. So, although morphologically it is isogamy, but physiologically it is called anisogamy. One thing to next, what happens next? So here you will get the gametic union. Eventually, one filament is found free, in the other zygospore is found. Why it is called zygospore? Because it is produced by means of conjugation. Clear? This is how conjugation. So what I said here, most important, the conjugation is pyroglycan. Conjugation conjugation is pyroglycan. And what I said here, it is morphologically isogamy, but physiologically anisogamy. This is what about the specific thing you need to remember this thing. Why it is called so? In Spirogyra, the conjugation is found to be morphologically isogamous type and physiologically anisogamous type. As the gametes are similar in size and shape in both the filament, so it is called morphologically isogamy. But as one is motile, other is non-motile, so here physiologically they are found different. That's why it is called physiologically anisogamy. Clear?
Sir,
Bolo.
so which one is motile?
Which one is motile means here, which one is motile you see? The blue one is showing more motile here. Any filament, anyone can be motile. If it is motile, then it moves towards this. But in one filament, it should be motile, and other it should be non-motile. Are you getting me?
Yes, sir.
So this is what about the conjugation in Spirogyra. Now I am showing is to in some variation some different structures in Kara, another LP. Kara is a green algae. So what happens here? It appears like this. This Kara, and due to its shape, is also called aquatic horsetail. What it appears like. This is one. The Antheridium, palms are produced. So eventually, sperms are released in the water. They are flagellated, will reach the ogonium and fertilizes the non-motile or sessile egg there to form the zygote. And in this way, fertilization takes place. And this type of reproduction is called ogamy. And so this is about the Kara, where you will get two different male and female reproductive structures. That's why Kara is also called aquatic horsetail due to its shape. Is it clear to you? So this is what about the different types of green algae. Now we come to the brown algae type. The Muchidi. Kara.
স্যার ওই ফিমেল অর্গানটার নাম কি
Ogonium.
আর মেলটা
WOGONIUM দেখা যাচ্ছে না? এখানে দেখা আছে
হ্যাঁ স্যার আর মেলটার নাম
অ্যানথেরিডিয়াম, তারা ব্ল্যাকে লিখে দিচ্ছি। ঠিক আছে
স্যার
স্যার ওরা তো গ্যামেটস জলে প্রডিউস করে তারপরে ফিউজ হয় না?
Actually, gametes are produced, those male gametes are produced within the antheridium. When the antheridium wall is broken down, the male gametes are then released into the water. By swimming, they reach the ogonium, and where inside the ogonium, it fertilizes the egg. বুঝতে পেরেছিস?
ওকে স্যার, ওকে স্যার, ওকে স্যার।
Now, we have presented the other type of algae. We have to proceed towards the brown algae. In case of brown algae, you know, it's also called Phaeophyceae. This is called Phaeophyceae.
স্যার বাউল
Brown spelling?
হ্যাঁ বলে দেখা যাচ্ছে?
Brown না brown?
Oh, sorry. Brown algae or Phaeophyceae. The next group. They are first of all, photosynthetic pigments. Chlorophyll A, chlorophyll C, and fucoxanthin. The most important that actually found in they are habitat marine. That means salt water. That is marine.
Sir, what you have written in the third number?
Fucoxanthin, FUCOXANTHIN. Is a yellow pigment. Evidence also a third one. Cell wall is made of an algene compound. Cell wall is made of cellulose and algene compound. Is it visible now?
Yes, sir.
Yes, sir.
So this is called rhizoplast, or it's called hold fast. Sorry. Type front, and each one is called blade. So these are the basic structure found in brown algae. The body is a hold fast by which it gets attached to the substratum. Stipe is the stalk-like structure that bears the photosynthetic organ, which is actually the front. Obviously, they are autotrophs and they can prepare their own food. But remember this thing, although in the beginning what I said, the flagella are found in brown algae which are unequal and lateral. I discussed it in the beginning. But you know, in the main plant body, flagella are not at all found. Flagella are only found in gametes. Whenever gametes are formed, you will get the flagella. During sexual reproduction, gametes are formed, and these gametes are found to be flagellated. And in these gametes, the flagella are found to be two unequal lateral flagella. Gametes are pear-shaped, having two unequal pear-shaped flagella, and asexual reproduction takes place by zoospores. They are also pear-shaped, having two unequal lateral so in brown algae, reproductive structure, flagella are found. So in brown algae, in reproductive structures, flagella are found. In zoospores, the asexual spores, and also in the gametes. Clear? Reproductive structures means zoospores, oblique gametes. Clear? So this is a so this is also important. Bladder, air bladder containing algae. Algae and important for used as
Sir,
wait. Source of manure and algae. হ্যাঁ বলো
sir, Dole laminaria belongs to Pteridophyta.
না this is the exception. That's why I'm writing here. What I said is most algae blood cycle is haplontic, but there is the exception. In Fucus, life cycle pattern is, you need to remember, diplontic is the exception. In Laminaria, it is haplodiplontic, although it is an algae. In Fucus, although it is an algae also, here the life cycle pattern is diplontic. Is it clear? Then,
Yes, sir.
so next one is the sargassum, used as foreign manure mainly, and next one is Ectocarpus. It is also used as fodder and food. So here also haplodiplontic life cycle is found. In both Laminaria and Ectocarpus, haplodiplontic life cycle is found, and in case of Fucus, you need to remember diplontic life cycle is found. So this is what about the what is called brown algae. Bolo.
In Sargassum, life cycle pattern.
Life cycle pattern haplodiplontic again. In Sargassum also.
In Sargassum also.
Sargassum also. I have to check again. Okay. It shows diplontic life cycle. Sargassum shows diplontic life cycle. Brown algae. I think it is clear now about the brown algae. Now we proceed towards the next one, the red algae.
স্যার ফুকুসের লাইফ প্যাটার্নটা কেমন?
Fucus.
হ্যাঁ
Fucus. Diplontic.
হ্যাঁ
Diplontic
Okay,
life
sir.
cycle. Now you see Rhodophyceae. Photosynthetic pigments. So phycoerythrin is responsible for characteristic red color of the red algae. Phycoerythrin is responsible for characteristic red color of red algae. Chlorophyll A is the universal pigment and chlorophyll D is there. Now, here in case of red algae, Floridian starch. Red algae, the basic food is Floridian. Reserve food is Floridian starch. And cellulose is made of cellulose, pectin and polysulfate esters. Then comes to the flagella are not found. Flagella are not at all found in red algae. Remember this thing. So another thing important in case of red algae, that here some specific species of red algae found, like suppose Glaciaria, Gracilaria, the most important one. Agate is produced. Gracilia is a red algae from which agate is produced. Okay? Remember this thing, the most important thing, Gracilaria, Gelidium also. Next one is contrast, most important one. So Gracilaria, one red algae, Gelidium is the other red algae. Do you know what is agar? Do you know what is agar?
Sorry, underwater plant.
Agar, actually no. Actually, it's a product which is used in making medium in the lab where different organisms can grow. Agar medium is used in the lab. This agar is extracted from both Gracilaria and Gelidium. So, this is the agar, actually the products of these two red algae. One is Gracilaria, other is the Gelidium. Is it clear now?
Sir, used in lab for?
For making medium, synthetic medium for the growth of or for growing different microorganisms. Used in the preparation of synthetic medium, agar medium, for growing different microorganisms.